Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we started a discussion on biases in RNA sequencing data. We talked about different types of biases and we also discussed some of the methods and tools to correct for those biases. And then we started talking about normalization. So, data normalization corrects for technical variation that is present in the data. Of course, it cannot completely eliminate technical variation, but the goal is to minimize the technical variation so that we can study the biological variation. In this class, we will continue our discussion on data normalization methods. We will introduce more and more sophisticated methods for data normalization, and this class and the next class we will talk about different types of normalization. For example, I have introduced, we will talk about within sample normalization, then we will talk about between sample normalization when we have multiple samples within a data set, and then finally we will talk about different types of data sets, right, combining different data sets and then doing something called batch correction. So, today's agenda will be first we will talk about normalization for read length and sequencing depth, and then second we will talk about between sample normalization, at least some of the techniques for between sample normalization. Here are the keywords that will come across RPKM, FPKM, and we will also see TPM. So, let us begin with the same diagram that we used in the last class. So, here we have these three genes, okay. You can see this gene A, gene B and gene C, and you have reads mapping to these genes, right. So, these reads are shown in blue and they are mapping to this, which we can convert now to the count, right. So, we have this read count, simple read count will give A. For A we have 6 reads, for B we have 3 reads, and for C we have 2 reads. And we also talked about gene length bias, right. So, the genes are of different lengths. So, I have also mention the size of this gene before, right. So, the one of the questions that we talked about is that does gene A have higher expression compared to gene B in this sample? Can we answer that question right from this data that we have? So, the answer is no, right. We need a lot more data, a lot more methods to actually analyze this properly, so that we can get an answer to this question. Okay. We also introduce another example where we are comparing among different samples, right. So, here we have sample 1, sample 2 and sample 3. We have these 3 genes again, and we have corresponding read counts, and we also have the total reads, right. So, we also talked about, right, we have this gene length bias and we also have to look for sequencing depth, right. So, each sample will have its own sequencing depth that will be different from other samples, right. This is common across all RNA-seq experiments, because you cannot always ensure that you will get exactly same number of reads for each sample. So, two questions that I introduced in the last class, right. So, does gene A have higher expression in sample 2 compared to sample 1, right. This is again dependent on a lot of factors which will actually analyze today, and we will see that if we can answer this question in this class. And then we have the second question. So, do genes B and C show lower expression in sample 3 compared to sample 1. So, again we will see if we can answer that question in this class, or maybe in the next class we will have more sophisticated tools which you can utilize to answer this question. Okay, so, the first question we will answer is how can we normalize raw count data for gene length and sequencing depth, right. So, we introduce this gene length bias because genes are of different lengths, and if you have a long gene, it is likely to have more fragments generated in the library, and hence you will get more reads for that gene, right. So, this is what we call gene length bias, and then we also have sample specific sequencing depth variation, right. So, each sample will get different total number of reads, and that can again influence the outcome, right. So, if you in a sample, if you have more number of reads, it is very likely that all the genes will also get more reads by chance, right. So, again we would have to do something about that, right, this variation in sequencing depth, so that we can actually do a fair comparison. Okay. So, this is the idea behind normalization, and the first question is, can we normalize for gene length and sequencing depth. Okay. So, let us start with a very simple normalization which actually normalizes for sequencing depth only. It is called counts per million, okay, or in short CPM. and then this is the very simple formula. Okay. So, you have normalized count for a gene or transcript. So, here the normalized count is calculated from the raw count using this formula. So, you have raw read count for the gene or the transcript divided by the total sequencing depth into 10 to the power 6. Okay. So, this is counts per million, right. So, we have to multiply with 6, 10 to the power 6. Okay. So, let us do this operation here. So, we will go slowly and so that you understand all these operations, and this will be actually helpful as we go into more complex operations. Okay. So, we will start with the same example, right. We have these 3 samples and we have these read counts for these genes A, B and C. Okay. So, the first step is we divide by the total sequencing depth. So, the total is there, right. So, we divide everything. So, for sample 1 the total is 11. So, we divide this column by 11. For sample 2 the total is 22. So, we divide by 22. For sample 3 it is 11 again, and we divide by 11. Okay. So, this is just an example with a very small number of genes, okay, and we do the same thing for real data sets where you have thousands of genes. Okay. So, then once we have divided, right, we just multiply with 10 to the power 6, okay, and you get this kind of answers. Okay. And what you notice here, the total, right, it comes to 1 into 10 to the power 6 for each sample, right. So, that is the idea, right, that is what we want to do. We want to normalize for the sequencing depth that was variable between these 3 samples. We have normalized for that and we have reached the same total sequencing depth. Okay. Now, one of the questions you might have is why are we multiplying with 10 to the power 6? We could have just left it like without this multiplication, right, the sum would be just 1, right. It is just a convention, okay, because when you deal with the real data sets, we have millions of reads coming for each sample, right. So, if you look at the read data, total read for a real data set, it will be in the order of millions, or nowadays it would be even more, right, in billions. Now, what will happen is if you divide the count for each gene by that big number. Okay. So, if you have, let us say for gene A you have count maybe 1000, for gene B maybe you have count have probably 100 or maybe less, right, and that number you divide by let us say 20 million. Okay. So, what will happen is that this number that this normalized number that you will get just by this division will be very, very small, right. As you can imagine, if you divide 100 by 20 million, that would be a very small number, that would be a fraction, right. So, to get something that is easily readable and also we can handle that better, right. So, that is why we multiply with 10 to the power 6, right. So, we get something like an float or very close, at least some integer values are there, etc., right. So, we have something like 1 to something like that, okay, and instead of very small fractions which it reads like 0.000 something. Okay. So, that is the idea behind this multiplication. Okay. So, that is what we have answered, right, why do we multiply with 10 to the power 6, so that we get numbers that are easily readable, right, for us. Okay. So, this is a very simple normalization. So, of course, it has a lot of drawbacks. So, one of the major drawback is that we are not doing any normalization for gene length. Okay. We are only normalizing for the total sequencing depth. Okay. So, as you can see, we have got the total same for all the samples, but we have not touched or worked with the gene length at all. Okay. So, that means again we cannot really compare expression levels of genes even within a sample. Okay. This is one. The second is we have not addressed the RNA composition bias. Okay. So, what is this RNA composition bias? We briefly mentioned in the last class. Okay. So, if you look at sample 3, right, gene A, the read count is 0. Okay. So, let us imagine that the expression of gene A has dropped, okay, and it is so low that we cannot detect it by sequencing. Okay. So, it is set to 0. What it means is that the total number of reads that are present, that are available for sample 3, those would be distributed between gene B and gene C. Okay. So, now what will appear is that gene B and gene C, they are showing higher expression in sample 3 compared to, for example, sample 1. Okay. So, this is the RNA composition bias. The read count for gene B and gene C, they are affected because the expression of gene A has changed. Okay. So, what you probably can now guess is that the read count for a single gene is not just dependent on its expression, but is dependent on the expression of the whole transcriptome. Okay. So, that is how the whole thing works. Okay. So, this composition bias we have not addressed, right. So, if you just look at this CPM data, this normalized count data, what you see is that sample 3 has higher count for gene B and gene C compared to sample 1 or sample 2, right. And you might say, okay, these genes B and C, they are showing higher expression in sample 3 compared to sample 1 and sample 2. Okay. So, if you conclude that, that will be erroneous, right, because we have not addressed this RNA composition bias, right, that is occurring just because the gene A, its expression has gone down. Okay. It is maybe B and C, they have not changed their expression, but because of change in expression of gene A, the distribution of reads are such that their gene B and gene C appear to be highly expressed. Okay. So, this is something that this method cannot address, which means again we cannot really compare gene expression across samples. Okay. So, this is a method that we will not use at all, because this is a there is a lot of problem, lot of drawbacks, but I introduced you this method because this will be helpful, right, for the further discussion in this class. Okay. So, let us move on to the next part, right. So, again because we have not addressed this RNA composition bias, we cannot answer the second question in these two questions that I said, right. So, first question was, does gene A have higher expression in sample 2 compared to sample 1? Then the answer is no, right. So, as you can see, right, the numbers are exactly identical, because we have taken that sequencing depth into account, right. Now, we can say that they are not different, right. If you look at sample 2 and sample 1, you see, right, for expression of gene A actually exactly identical, the read count, normalized count data is exactly identical. Okay. So, the answer is no, but we cannot answer the second question, right, because we have not addressed the RNA composition bias, right. So, would genes B and C show lower expression sample 3 compared to sample 1? We cannot answer that question because we have not addressed this RNA composition bias. All right. So, let us now move on to the next normalization. Okay. So, this will address this gene length normalization as well as normalization for sequencing depth. Okay. So, the first one we will talk about is called RPKM or FPKM normalization. Okay. I will introduce these terms in a moment, what RPKM and FPKM means, and then we will also talk about the TPM normalization. Okay. So, let us start with RPKM FPKM normalization. Okay. So, RPKM stands for reads per kb per million mapped reads, and FPKM stands for fragments per kb per million mapped reads. Okay. So, RPKM FPKM, the method is exactly identical. The only difference is, instead of counting reads, we count as fragments. Okay. So, why do we count as fragments? Because if you are dealing with paired end data, reads do not make any sense, right. The fragments make more sense there. Okay. So, if you are dealing with single end data, you can use RPKM, but if you are using paired end data, you would have to use FPKM measure. Okay. So, the terminologies are different, but the normalization procedure, that is exactly same for both of them. Okay. So, let us now look into the normalizations. Okay. So, here are the steps of this method. So, the first step is we divide by the total sequencing length depth. Okay. So, we have this depth calculation. So, it is actually very similar to what we have discussed just before this, right. So, the CPM normalization method, we follow the same process, right. We divide by the total sequencing depth. Okay. Then we multiply with 10 to the power 6 as before, right, and you know the reason now, we understand the reason. Then we actually normalize for gene length, and to do that we divide by transcript length in kb. Okay. So, this is gene length or transcript length in kb. Okay. So, that is something you should note. Okay. So, the formula is like this, right. So, we get the normalized count for a gene or transcript. So, by dividing the raw read count with total sequencing depth as well as by gene and transcript length, and we multiply with 10 to the power 6. Okay. So, million, again we understand why we do that. Okay. So, let us take this earlier example, right, and let us now do this RPKM FPKM normalization, right. We follow this method. Okay. So, first we divide by the total sequencing depth. Okay. So, in this case, for sample 1 this is 11, for sample 2 22, for sample 3 it is 11 again. Okay. So, let us do that, let us do the division as before, right. We have seen this before. Okay. and then we multiply with 10 to the power 6, okay, and these are the numbers that we get. Okay. We did this before, right, just before this method. Okay. So, we get this total is same for all the samples. Okay. Now, what we do is we divide by gene transcript length in kb. Okay. So, you can see this gene length here, right. So, for gene A we have 2 kb, for gene B the length is 1 kb, for C is 0.5 kb. Okay. And that is what we do, we divide these counts that we got by this gene length in kb. Okay. So, divide by 2, divide by 1, divide by 0.5. Okay. And once we do that, right. So, we can calculate what would be these values and we get the total. Okay. And what you would notice now is that the total is actually not same for all samples, okay, and this has changed. Okay. And this is a problem, right. Again we are comparing a different number of total number of reads, right, or total sequencing depth. This is a transformed depth, of course, it has been transformed with all those division, etc. But again if you look at the total, this is different, right. For example, sample 3 is different from sample 1 and sample 2. Okay. So, this is a problem, okay, because then we are not comparing across samples which have exactly same sequencing depth, which we got earlier when we were doing this CPM normalization. Okay. So, this creates a problem because you cannot then compare across samples anymore. Okay. And also you cannot compare across samples because the RNA composition bias is still there. We have not done anything about that yet, right, because again if you see this number is actually higher, right, for sample 3, these counts that we got, normalized counts, this is higher in sample 3 for gene B and gene C compared to sample 1 and sample 2. And you might erroneously say, okay, gene B and gene C, they are highly expressed in sample 3, but that is probably not the case, again because gene A has changed its expression. Okay. So, what are the solutions then? Okay. We cannot answer this question correctly yet, right. We can answer, of course, but that will be an erroneous answer. Okay. So, let us look at the other normalization, which is a slight deviation from the RPKM FPKM normalization. Okay. So, this was proposed later after researchers found out the drawbacks of this RPKM FPKM normalization, okay, and it is called TPM normalization. Okay. So, the TPM stands for transcripts per million. Okay. So, very similar idea, but when you see the formula, you will see the formula is actually identical, but the steps are slightly different. Okay. So, what are the steps? So, here you first divide by the transcript length in kb. Okay. Instead of dividing by the total sequencing depth, you first divide by the transcript length in kb. Okay. Once you have done that, then you divide by the total count. I am not saying total sequencing depth because the total will change because of this division by transcript length, right. So, it is a total count that we will get after this division, okay, and then multiply with 10 to the power 6. Okay. So, you see like just one step, like this division by transcript length, this is the first step now instead of the last step in FPKM RPKM normalization. Okay. So, let us take this example again and see what we get when you do the TPM normalization. Okay. So, the formula is actually identical, as you see. We get normalized count for a read for a gene or a transcript by dividing the raw read count for the gene and or transcript divided by the gene transcript length and divided also by the total sequencing depth into 10 to the power 6. Okay. So, this is the formula that we also got earlier, but the steps are slightly different, the order of steps, right. Okay. So, let us take the example and let us do the analysis, do the TPM normalization ourselves with this very small data set, and we get an idea. Okay. So, here again we have 3 samples and we have the genes A, B, C, right, and for which we have the raw read counts. Okay. So, remember now the first step is division by the gene length. Okay. So, we will divide by this gene length 2 kb, 1 kb, 0.5 kb, right, and we will get some numbers. Okay. So, we do that, okay, and I have not written the total, we will get the total once we have done the division, okay, and you see after the division this is what we get. Okay. This is not the total number of reads, it is actually total some sort of transformed total, right. So, you have 10, 20 and 15. Okay. So, the next step now is to divide by this total, okay, that we have got for each sample. Okay. That is because the totals are different, right, we have to divide by this. Okay. So, once we divide by the total, what we get is, you can see, right, we have divided sample 1 by 10, sample 2 by 20 for each column. So here for sample 3 we have divided by 15, right. Okay, so we get the numbers, right, and as you can see, now the sum is 1 for all of them. Okay. So, just a simple tweak actually can change the whole thing. Okay. Sum is 1, the next step is multiply with 10 to the power 6. In this example, because the read numbers are very small, right, and we are dealing with very small number of genes, we are getting pretty good numbers, right, 0.3, 0.3, 0.4, etc. But in real scenario with real data sets where you have millions of reads, these numbers will be very, very small if you just leave after the division, right. So, 0.00 something. Okay, so, once we have multiplied with million, right, we see this total that is exactly same for each sample, right. So, that is a good thing, right. We have normalized for total sequencing depth. Okay. And now we can then compare perhaps these across samples, but the problem is we have not addressed the RNA composition bias and it is still there. Okay. So, which means again we will reach some erroneous conclusions because we have not addressed the issue that gene B and gene C they get more reads only because gene A shows lower expression in sample 3. Okay. So, this, to summarize whatever we have discussed so far, right. So, these are the drawbacks of CPM, RPKM and TPM normalization. Okay. We have discussed these methods and we have seen these are quite simple normalization methods. So, one of the problems with CPM normalization, it does not normalization for gene length, right. So, again, which means we cannot compare expression levels of genes even within a sample, right, because we have not taken the gene length into account. We have also seen that RPKM, FPKM normalization does not allow us to compare gene expression across samples because the total count is different, right. At the end of normalization, the total count that we get is different for each sample, right, and that means we have not done proper normalization, right, and also in addition you have the RNA composition bias that we have not addressed. Okay. And TPM normalization, it addresses these issues, right. We get the total count same for each sample at the end of normalization and we also normalize for gene length, but it still does not address the RNA composition bias issue. So, we cannot really compare across samples because we might end up with some erroneous conclusions. Okay. So, as you can probably understand, these methods, especially RPKM and FPKM and TPM, these methods are good for within sample comparisons. Okay. So, if you are comparing gene A versus gene C or gene 100 to gene 1000, right, that is probably okay with these methods. If the moment you go comparison want to compare across samples, then you will face some issues, right. You might get some erroneous results, some erroneous conclusions. Okay. So, what are the solutions, right? Okay. So, because as we have seen, this RPKM, FPKM, TPM does not really normalize for this RNA composition bias. So, what can we do to address these situations? Okay. So, the answer is, of course, we need to do something called between sample normalization. Okay. So, RPKM or TPM methods, right, they are not doing between sample normalization, they are simply doing within sample normalization, right. So, we do the gene length and also divide by the sequencing depth. So, this is within sample normalization and we are not comparing across samples within a data set. So, this becomes very important in many analyses that we do. Okay. So, one of the major applications of RNA-seq is to do something called differential gene expression analysis. Okay. We will talk about this differential gene expression analysis later on in the subsequent classes, where we actually look into the expression levels of the same gene across different conditions, across different samples. Okay. So, which means we have multiple samples within a data set and we are comparing expression levels of genes across these samples. Okay. So, to do that effectively, we need something called between sample normalization, right. So, we need to address some of the issues that we have just mentioned, right, RNA composition bias, etc. Now, we will talk about these two methods today. Of course, there are many more methods, more sophisticated ones that we will talk about in the next class. Okay. So, the first one is called the median normalization and the second is called upper quartile or EQ normalization. Okay. So, let us now go into this median normalization and EQ normalization and see, right, how the normalization is done between samples. Okay. So, again this will help you understand the more sophisticated methods in the next class. Okay, so let us talk about median normalization. Okay, so one of the assumptions in median normalization is that the read count distributions in the samples, they differ by a constant value, right. So, if you have seen these examples of 3 samples, right. So, the assumption here is that the distributions, they differ by a constant value, right, and we can simply scale these distributions to bring to the same median. Okay. So, once we start with this assumption, the goal becomes very simple, right. So, we actually scale the read counts by the median value of each sample. Okay. So, we calculate median and we scale the read counts by the median value. Okay. And at the end of the normalization, what you would see is that all samples will have the same median value, right. That is the idea behind this normalization, right. So, because of the assumption, the goal is simply to bring the median value to the same value for all samples. Okay. So, again if we go back, right, we cannot calculate of course a median for only 3 numbers, that will not be appropriate, right. So, again you can think about, right, how you are going to proceed, right, and then I will show the analysis with a real data set where you have 6,000 7,000 genes, and then you can probably guess, right, what will happen. Okay. So, let us just for a moment think about, right, how you are going to do this normalization, median normalization for these 3 samples that you see, okay, for sample 1, sample 2 and sample 3. So, what you have to do here is that you have to calculate the median value for each of these samples, right. So, you have this read count. So, for sample 1 you calculate the median value, for sample 2 you calculate the median value, for sample 3 you calculate the median value, and then you scale by the median value, right. So, you kind of scale each of these read counts with the median value that you get from each sample. Okay. So, this is the idea, right. Step 1, we calculate median for each sample here and then scale the counts by the median value of each sample. Okay. And at the end what you will get is that the median value for each sample will be the same. Okay. Before you start the normalization, of course, they are not same, but after normalization they would be same. Okay. So, what I will do now, I will actually show the results from a real data set with 6,000 7,000 genes, right, and we can then do the median normalization. Okay. So, that is what I am going to do with a real RNA-seq data set, and I will show the results in a plot called RLE plot. Okay. So, do not worry about this, it is called RLE plot, right. So, relative log expression plot. We will talk about this relative log expression calculation, right, a bit later, but it is a good tool to visualize our results. Okay. Okay. So, here is the raw count data for 16 samples before normalization. Okay. And the samples, okay, you can ignore the sample names, they are labeled in some way, but pay attention to the colors that you see. Okay. So, the colors, the samples that are of same color, right, they are technical replicates, okay, of the same condition or same sample. Okay. So, you have 16 samples and we have 4 conditions, okay, and for each condition you have 4 replicates. And as you can see, these replicates again show quite a bit of difference, right. For example, you can notice here, right, this replicate of this set, right, it shows quite a different distribution, right, for raw count. Okay. For now, again I have not explained what is this RLE, right. So, this you can ignore the y axis now, because this is a transformed axis, because raw count cannot be negative, right, you cannot see raw count is not negative. So, this is a transformed one, but we will use the same scale for all the plots that I show you. Okay. Now, once we do the normalization, okay, you see, right, because we have done median normalization, the median is same and is set to 0 for all the samples, right, because we have scaled by the median of that sample. So, the median should become 0 for all samples. Okay. So, this is after median normalization. Okay. Again we are doing again showing the RLE plot after median normalization, okay, and you see median values are same for all samples here. Okay. So, that is the idea behind doing this median normalization. There is a variant of this, right. So, it is called upper quartile normalization or EQ normalization. Okay. So, the assumption is sample distributions again differ by a constant value, right. So, again read count distributions, they differ by a constant value across samples, and we can simply scale these distributions and bring the upper quartile to the same value. Okay. So, hopefully you know what is upper quartile, right. So, here is the scaling the read counts by the value of the 17th percentile of read counts for each sample. Okay. So, we have median, here it is 75th percentile. Okay. So, again I will show you the results from the same data. Okay. So, here again is the raw data, raw count data before normalization, and this is after upper quartile normalization. You can see for some samples, okay, it looks pretty good, but for some samples here, for example, this one or this one, the median is not 0, okay, and that is expected because here we set the 75th percentile to the same value and not the median. Okay. All right. So, these are some of the methods that we can use. Now, there is a hybrid method that combines this FPKM with upper quartile normalization, right. So, in FPKM at the end what you got is the total count was different for each sample, right. And so, you can address that if you do something called an upper quartile normalization, combine that with UQ normalization, right, and you can address this issue, right. So, here the upper quartile normalization is combined with FPKM normalization, and this helps us in a better comparison of gene expression levels within and between samples, okay, because we are doing an within sample normalization, which is the FPKM, followed by an between sample normalization, right, which is the UQ normalization. And this has been done for many data sets. You will find many public data sets where the count data that is given is sometimes referred as FPKM UQ data. Okay. So, once you see this kind of data, you will probably then understand, right, what has happened, right. First normalization was FPKM, then there was a UQ normalization. Okay. So, what are the drawbacks, right. So, we have talked about some of the between sample normalization, and one of the first drawbacks, right, is that the assumption that sample data vary by a constant value, it does not hold for most cases. Okay. It is not just simply the distributions are shifted and the median should be brought to the same value. This this does not hold for most samples. Okay. And also we have not addressed the issue of RNA composition bias here, right. So, we have not touched upon that at all when we are talking about this between sample normalization here. Okay. So, here are the references that we have utilized for this part. So, to summarize, we have looked into normalization methods for gene length and total sequencing depth. We have talked about some of these methods, right. So, FPKM, RPKM method, TPM method, right. And then we have seen, right, these methods are good for comparison of experiment within samples, but they are not suitable for comparison between samples, right. So, we have explained the reasons why that is the case, and then we have talked about normalization between samples, also median normalization and UQ normalization. So, these are only two methods that we have discussed, but again they have not addressed some of the issues that are still remaining, and that is why we need better methods, which we will talk about in the next class. Thank you.